Well, thanks for that lovely introduction, Dagmar. It's a great pleasure to talk here always. And for those of you who saw my last public lecture at Cosmic Hub, which was about a year ago, at the Plant Biology Symposium, this is going to be a sort of update on where we were then. And I entitled it Send in the Clones Part 2 for that reason. So clones are very important in plant biology. I'm going to explain why, and then I'm going to tell you about the oil palm genome sequencing project, which I've been involved in actually for the last 10 years now, and some recent discoveries that we made. And then finally, I'm going to tell you about the potential for other sorts of plants as biofuels and perhaps to do something about climate change in the long term. Okay, so clones. Well, when we think of human clones, especially human reproductive cloning, it's obviously a very controversial matter. It's been the subject of many movies in the last few years. But clones, of course, are much, much more common and widely propagated in plants. In fact, the word klonos in Greek means a little twig or branch that you can propagate as a vegetative cutting, which will completely recapitulate the genetic material found in the parent tree, and so effectively as a clone. Clones are actually very, very common in nature. The oldest and probably the largest organism on Earth is a clone. It's a stand of quaking aspen in Utah. It's called Pando, which is the Latin for I spread. And it's estimated to be about 50,000 years old. The entire clone is male, so it's an interesting life habit. And it weighs, at the moment, with the 40,000 or so stems or ramets, it weighs something like 6,000 tons. Over the years, it's expanded over many, many acres, and actually it's thought to be older than the last 2 ice ages. It propagates through the roots, so the roots are the way in which these stands are related. But of course, over time, as well as being entirely male, Pando has lost most of its fertility, and so it probably hasn't sexually reproduced in something like 10,000 years, which is, as I said, an interesting life strategy. And there are other clones, plant clones, that you're probably more familiar with than Pando. Grapes are propagated as clones. They're propagated as cuttings, actually by grafts, and they can occupy huge areas. This is a vineyard in Chile I was at fairly recently. And of course, each variety of grape is highly prized for its uniformity. So if you have a Pinot Noir, you want it to be Pinot Noir. Pinot Gris, you want it to be Pinot Gris. But in fact, many of these prized clonal varieties were derived from each other, and that's because they can be unstable. And so, for example, Pinot Blanc is actually a derivative of Pinot Noir, and it's a derivative that arose through vegetative propagation, probably sometime in the Middle Ages. And we now know a lot more about this variation. We sometimes call this epigenetic variation between otherwise genetically identical clones, between individuals that are clonally related. Much of this variation in clones is caused by epigenetic effects on something called a transposable element. These were, of course, discovered here at the Cold Spring Harbor in the 1940s and 50s by McClintock. And they can cause actually genetic as well as epigenetic changes in clones using mechanisms and principles that she discovered a long time ago. And in fact, those varieties of grapes that I just showed you turn out to be caused by the insertion of these transposable elements. So transposable elements are responsible for much of this variation. So Pinot Noir has this beautiful dark color anthocyanins in the skin. Whereas Pinot Blanc has this insertion of a transposable element, or sometimes called jumping gene. These are pieces of DNA that can move around the genome, and when they integrate next to a gene or within a gene, they can actually change the way that gene behaves. A subsequent rearrangement, in which most of the transposon was lost, has led to Pinot Gris. And so the full range of color that is in these Pinot grapes can be explained by just the sort of variation that Barbara was talking about. Okay, so why are clones successful in nature? Considering the fact that they often sacrifice fertility, as Pando has done over the last 80,000 years, but they still survive very well, and each of their individual members survives. And part of the reason for this is that they maintain something called heterozygosity. What that is the genetic contribution that comes from both the original parents. This is something you're probably very familiar with. It's often called hybrid vigor. And hybrid vigor is really the key to much of modern plant breeding. Hybrid vigor is still not fully understood. There's a lot of theories about hybrid vigor, and I'd be delighted to tell you that we think we're getting a little bit close to at least one of them, even in the oil palm. But it's still one of the great mysteries of plant genetics. But this is why clones, which of course maintain that hybridity, because they don't sexually reproduce, they don't shuffle the genes, the combinations from the parents, they just maintain them from one propagated propagule, as they're called, to the next. And in contrast, of course, sexual inbreeding, which is practiced in many plant breeding schemes, leads to poor performance. It leads to something called inbreeding depression, and actually that's true of inbred animals and inbred humans as well. Just think of the British royal family, for example. I didn't really say that. You know, the success of clones really results on this maintenance. Well, the principles of hybrid vigor were actually first elucidated again here at Cold Spring Harbor more than 100 years ago now, in 1908, by George Schull, who perfected the art of inbreeding parents of corn and then crossing them together so that they generated a uniform hybrid population. Now, those inbreds weren't particularly high yielding, they were just stable, and that was difficult to do, but he did it. But the hybrids were very high yielding because, again, they got both contributions from both parents. And as I said, at least in maize, the molecular, the sort of DNA basis for this hybrid vigor is still not fully understood. But working here, as well as in Israel, before with his former advisor, Danny Zamir, this is Zach Lippman, who's an assistant professor in the plant group, and he was able to show that one of the most important genes involved in tomato breeding is called the S gene, is in fact one of these genes that exhibits what's called single gene heterosis, hybrid vigor, in which just a single allele at each gene is enough to give you a heterozygote, a hybrid, that's better than both parents, and way better. So these hybrids are used in commercial breeding, and they produce a lot more yield than other tomatoes. So this is a very important discovery made a few years ago. Okay, so what I'm really going to be focusing on today is how the principles of plant genetics, what I told you about clones and hybrids and so on, can potentially be used to solve some of the major problems in agriculture, and also, importantly, in bioenergy, in the impact of bioenergy on, for one thing, providing energy, which is itself valuable, but also solving one of the great crises of today, which is, of course, the carbon problem. You probably don't need me to remind you of this, but essentially fossil reserves, which are essentially ancient plants and ancient animals, which have contributed carbon to these fossil deposits, are consumed in the most crude way by burning to produce energy. And that energy, unfortunately, also gives rise to the release of atmospheric carbon. Atmospheric carbon would normally, through various mechanisms of climate change, have some pretty nasty effects. But plants can fix that carbon. They can use photosynthesis and reduction power that comes from photosynthesis to make sugars initially out of that carbon. And that fixes it, it takes it out of the atmosphere. So if you can use plants, existing plants, instead of fossilized plants as fuel, you could potentially close this circle and prevent the release of excess atmospheric carbon. So that's the real promise of biofuels, in addition to issues like, oil dependence and economic issues. This is really the much more long-term value of biofuels. Okay, well, if we look at what sort of crops have been used for biofuels, there's a number that have been used. I'm just giving a little taste of them here. One of the most popular in the United States has been corn itself. But actually, corn has, of course, been bred and domesticated actually over thousands of years and bred over hundreds of years to produce food, not fuel. And it's in fact not that efficient at producing fuel. And the reason for that is you need a lot of high inputs, so fertilizer, you need to carry it around, You need decent land, irrigation, and so on to make high-yielding corn. And we have very high-yielding corn. But after you've put all that energy in, the amount of energy you get out is, at best, a ratio of about 1.5, which isn't very good. And that's actually a generous estimate. Some people think it's actually less than 1. Oil crops are often better than that. So rapeseed oil, for example, is decent at around 2 or 3 or so. But really, to get up to the really high-value energy crops, we need to look at much more recent or recently domesticated plants, such as sugarcane, which is one of the biggest biofuel crops in the world, palm oil, which I'll be telling you a lot more about, and some new technologies that I'm not going to tell you about, in which we're trying to directly convert cellulose material, think of paper if you like, but the stems, the stover of plants, into ethanol. This has not yet been done at any sort of efficiency that makes sense, but industrial plants already exist. So this is a potential hope for the future. But right now, the most high-yielding energy crops are in fact clones, or are propagated as clones. That includes oil palm trees, as I'll explain in a moment, but also probably more familiar sugarcane plants. So in Brazil, sugarcane has been a very effective source of biofuels. Of course, the way that works is you get the sugar out of the plants, you then convert it into ethanol using fermentation and use the ethanol as a liquid transportation fuel. You just add it as an additive to gasoline. And in fact, sugarcane is propagated as clones. The way that's done is as vegetative propagation from year to year. You can plant a little piece of the stem. You can't actually do that for every year. It only lasts about 6 or 7 years, and then you have to go back and replant. But nonetheless, you get these very uniform and very high-yielding populations of clonal sugarcane. And sugarcane, of course, can only be grown in certain parts of the world, but nonetheless, in those parts of the world like Brazil, it's a very important part of fuel production. But oil palm is actually, right now, the most efficient producer of bioenergy on the planet. For every unit of energy you put in, you get something like 9 units out. And furthermore, it comes out as biodiesel, which is an extremely efficient fuel that you can easily convert into jet fuel and so on. And in fact, Richard Branson flew one of his planes on palm oil to demonstrate this about 5 years ago. And there's been a lot of interest in palm oil as a potential biofuel then because of this. There are numerous problems with palm oil, and I'll be getting into that in a big way in a moment. But anyway, it was a number of years ago now that we started our collaboration on oil palms. I've got a lot of pictures from my various collaborators around the world. I hope you don't mind. I'm not a terrific cameraman, but you get the idea. The biggest and nicest collection of palm trees I know of is in the Botanic Garden in Rio. And this is the Royal Palm Avenue from that garden. And it just gives you an idea of the sort of tremendous growth capacity of these wonderful plants. There are many species of palm, but it's the oil palm trees that really give rise to all that oil. Palm trees are actually monocots. This is a branch, an order of flowering plants. So flowering plants you'd be very familiar with. And monocots actually include the grasses, so corn, wheat, sorghum, and sugarcane are all monocots. And so are palm trees, and they've just been placed on this evolutionary tree, which was actually constructed from DNA sequence in a collaboration with some very good friends of mine at the Museum of Natural History here in New York, who collaborated with us on the oil palm project as well. And they were able to place oil palm molecularly as botanists had before, which is good, so we all agreed, in a little clade here of tropical crop plants, which include ginger and turmeric, funnily enough, which you would have thought didn't look anything like palms, right? But from a DNA standpoint, from an evolutionary standpoint, they're actually very closely related plants. So oil palm trees then are in this genus called Elaeus. And they come in a couple of different species, as I'll tell you in a moment, which turned out to be very important for the genome sequencing project, which we began, as I said, about 10 years ago now. I will first tell you about those 2 species. So the 2 species are Elaeus guineensis, which is the African oil palm. So it's native to West, Central, and East tropical Africa, Sub-Saharan Africa. Whereas Elaeis oleifera is found in Central and South America, and so, for example, in Brazil. Both of them coexist in tropical rainforests. It's their natural habitat, or at least related tropical niches. And they are surprisingly interfertile. The reason that's surprising is that from an evolutionary standpoint, new and old world plants separated usually at the time when the two continents separated, which was about 60 or 70 million years ago. The two continents were fused at one time, called Gondwana. And so one of the interests of sequencing the genomes of these plants was to find out how on earth that could have happened, that these two species could have retained this ability to still cross to each other. And one idea, of course, was that, in fact, they're much more closely related to each other than you might imagine, and that, in fact, one migrated to the other, most likely from South America to Africa, or perhaps the other way around. And one reason that that's a popular idea is that if you look at the palm tree lineage, here are date palms. In northern Africa, of course, there are many, many species of date palms, and I think they're all, almost all, from the Old World. But oil palms are actually quite closely related to coconuts. As you'll see in a moment, that turns out to be important for the gene that we found called the shell gene. Think of a coconut shell. So one idea then, of course, coconuts can essentially propagate anywhere in the world because they float on water and they could just float anywhere. And so that's why coconut trees are all over the beaches in the South Pacific and so on. So one idea then was that oil palms might have been derived from some ancient coconut that had been able to propagate in that way. Turned out not to be true, just to spoil the suspense, but it was a good idea. It was actually my favorite theory at the time. Okay, so as I said, we established a collaboration about 10 years ago now, headed by the Malaysian Palm Oil Board, which is based in Kuala Lumpur in Malaysia, a place that I spend quite a bit of time, as well as Orion Genomics, which is a Cold Spring Harbor spin-off company, Cold Spring Harbor itself, of course, and our friends at the American Museum of Natural History. Between us, we were able to sequence the oil palm genome. It's big genome. It's almost 2 billion bases in size, 16 chromosomes, and it's highly repeated and doesn't really have any what we call reference that you can look at. You have to assemble the whole thing like one huge jigsaw puzzle in which most of the pieces are identical. And so it's an extremely difficult genome to sequence, but we did it. And we were able to publish it very satisfyingly in Nature earlier this year in July. And at the same time as publishing the genome sequence, we were also able to identify the single gene that has the biggest effect on yield of oil. That's the amount of oil you get per tree. You can also think of it, perhaps more provocatively, the amount of oil you get per acre of rainforest that has been sacrificed to allow growth of these trees. And so it's a very important thing to get at this oil yield. And finding that gene is quite a fun story. This is going to be the scientific part of the talk for those of you who don't want to take in too many details. I thought I'd throw in some real science just to give you an idea of what that's like. Before I do that, this is the Malaysian part of the team. Actually, these are the guys from Orion Genomics, and this is the leaders of the Malaysian team, including Dato Chu, who's actually the Director General of the Malaysian Palm Oil Board. And we really had a great deal of fun with these guys. They're amazing, an amazing group. And this is Rajinder Singh, who's the first author actually on both papers. Okay, so the genome. As I said, it's 1.8 gigabases in size. That's an awful lot of zeros. And is equivalent in size to the human genome in size, and actually worse in terms of complexity. There are 16 chromosomes, and we estimate from informatics something like 38, maybe 40,000 genes. So in that sense, it's quite similar to the grasses that we know so well, like rice or maize. And actually, the maize genome is similarly difficult and complex and difficult to sequence. As I said, there are only 2 species known, which is unusual for palm trees and quite unusual for angiosperms in general. And perhaps it's the fact that there are only 2 species which allows the interfertility, which I'll get in a moment. We also discovered that palms in general have these ancient duplications where one chromosome looks a lot like another. And these ancient duplications, and only duplications, not triplications, suggest that the genome as a whole was doubled something like 30 million years ago, leading to both date palms and oil palms. Sorry, 60 million years ago, leading to both date palms and oil palms. So we discovered all these genes. There are lots of nice chromosomal features. It's a very satisfying genome. Importantly, of course, we wanted to find out what was going on with oil in this genome, and I'll get to that in a moment. But first, I'll tell you the evolutionary result, which was a lot of fun and turned out to be the title of the paper, in fact. So our friends at the museum in New York were able to use the genome sequences or what's called transcript sequences of more than 150 species of plants worldwide. These have been collected in computer databases over the years, and we're able to get a really accurate picture of the evolution of all of these species. And when they did that, they found that the African and American palms were separated by about the same distance as other Old World and New World crops. For example, here's sorghum, which is an Old World grass, and zea mays, which of course is a New World grass, and they're actually closer together than the 2 oil palm species are. And for those of you who are plant biologists, of course, you can't successfully get fertile hybrids from maize and sorghum. You can't cross these together and expect to get anything that looks like a fertile and successful plant. Whereas oil palm breeders are actually using the interfertility of the American and African palms in breeding schemes. So it's quite extraordinary that oil palm trees have remained fertile for all that time. 60 million years, that's a really long time. And there's actually a theory that says that the fewer species you have, the more likely that is to have happened. So if you think about it, if there's only 1 species in each continent, they never see another species, so there's no need to build up reproductive isolation. So that was a very interesting result. We also, as I said, we looked at the genes responsible for oil biosynthesis, and we compared those genes in oil palm to date palm and also to banana, which is recently sequenced by an African and South American consortium. And when we did that comparison, we found, perhaps not surprisingly, that genes responsible for oil biosynthesis are overrepresented. There's more of them. There's more types of them, which probably means they're actually used in more different tissues of the plant. And if you think about it, the fact that oil palm produces all this oil in its fruit is its unique defining feature. Most plants only produce oil in their seeds, not in their fruit. And so this made a lot of sense. And I'll get back to that point again in a moment. Okay, but we were also able to take advantage of the extraordinary genetic resources that the Malaysians had built up over the last few decades. And actually, much of this work was started by Belgian breeders in the Congo before oil palm was introduced into Southeast Asia, which was something like the late 19th century, actually mostly as an ornamental palm, and only really used commercially from the 1920s onwards. And it was in the 1920s that a Belgian breeder working in the Congo realized that there was an example of single gene heterosis, exactly the same sort of phenomenon that I told you about Zach Lippmann finding in tomato was happening in oil palm. And the gene responsible actually had a name. The gene is called the shell gene, or SH, and it's responsible for the thickness of the shell. So I told you that oil palm and coconut are quite closely related. If you slice open an oil palm fruit, either one way or the other, you'll notice that inside is a white endosperm, looks a lot like coconut flesh, and it's surrounded by this thick fibrous shell, which is the equivalent to that awful thing that you need a machete for to get off to eat a coconut. And of course, it's much smaller, but it does decrease the amount of oil because it occupies so much of the surrounding fruit. Now, what this breeder realized was that commercially grown and actually a large number of wild accessions of oil palm were of something called the tenera, or thin-shelled form, and they could be produced by crosses between the thick-shelled and a rare type of oil palm called the Passifera, which didn't have a shell at all. And he was able to trace this to the single gene by classical genetics. In other words, it segregated as a single gene, which he called shell. And he realized then that the Tenera palms, which are thin-shelled, were heterozygous. So they got the alleles from both parents, and so they were hybrids. And those hybrids yielded something like 30% or even more oil than either parent. And part of the reason for that was the Passifera ones, which you would have thought might yield a lot of oil, are actually mostly sterile, female sterile. So you can only use pollen from these plants, and that's why they're so rare in nature. Okay, so given that these genes were discovered in the 1920s, they've been used genetically for breeding oil palms since breeding began, mostly after World War II. So this is just a summary of what I just told you. Oil palms exhibit this thing called single gene heterosis, in which the amount of oil that you get out of the mesocarp, that's the fruit, is much higher, and actually it has much higher quality because the shell is thin enough to allow accumulation of that oil and thick enough to allow fertility. You have to have both. It's sort of like the Goldilocks hypothesis, if you like. You know, it's just right. And the most productive bunch composition then, these fruit bunches, and you really do get a huge amount of oil. I mean, weight for weight, you get something like 40% of the weight of one of these bunches after steaming is just oil. It's remarkable. This meant, of course, that these Passifera palms, which I said were quite rare in nature, have this recessive shellless allele. What that meant is that we could take advantage of the extraordinary breeding that had been going on in Malaysia since the 1950s to find the shell gene. I just sort of give this little atmospheric photograph here of a shell, of an oil palm plantation in Malaysia. You can see sort of how dark it is and how mysterious the shell gene was. It had always been the sort of holy grail of biotechnology for the Malaysians. So what they had for us were pedigrees. They'd been maintaining pedigrees from their breeding schemes for decades, going back to the 1950s, where some of the earliest palms, and amazingly enough, of course, these are quite long-lived trees, so many of these trees are still around. You could still get DNA from them, for example. And they developed these pedigrees where they crossed together palms of differing allele at the shell gene, so they're either Tenera or Passifera. And then we could select some of the more recent versions of this from this pedigree, in which the genes had all been shuffled up enormously as much as you could, and try to find the shell gene in those palms. And we did that using something that we could only have done in the last 5 years or so, which is something called next-generation sequencing, in which you can, once you've got a good quality sequence of the plant you're interested in, you can then sequence many hundreds of other plants using this very high-throughput instrumentation that allows you to get all of the variation that's found in those plants from the original reference. And when we did that, we used a technique called homozygosity mapping. Again, this is for the cognoscenti, but it was fun because it had been first proposed by Eric Lander and David Botstein in the 1980s, before this technology was available. And they'd proposed it as a way to find actually human diseases that arise in inbred human populations. And so this was a perfect opportunity to use this using next-generation sequencing. And so within the rough genetic interval where we knew the shell gene had to be, there was just one region that was homozygous. That means there's no variation. This is just plotting variation here along the genome. Sequencing just about 50 trees was enough. And when we compared this to the genetic interval, there was literally one gene that was in that overlap. And that's amazing for those of you who do positional cloning in any organism, to get that sort of resolution. And this is what we can do with these next-generation sequencing technologies. So this was really applying the cutting edge of DNA technology to the Malaysian problem. So the shell gene was very satisfying in terms of what it encoded. It encodes a gene, which I'll tell you in a moment. It's already known to have a similar function in other plants. But what was really neat is it turned out that the Malaysians who collected something like 10,000 accessions of oil palm from Africa, so they'd gone out on collecting expeditions over the last few decades, actually led by an incredible guy called Dr. Rajan Naidu, who must be in his 70s, but still goes out there and climbs into compounds and takes this stuff. He's amazing. But anyway, it turns out that they'd collected two different alleles of the shell gene, which they'd used in their breeding, and actually had shown unwittingly controlled the same phenotype because they crossed them together. And these two mutations were only 6 base pairs apart in the genome sequence. And remember, the genome sequence is 2 billion bases. So the fact that we found these two mutations so close to each other is like finding 2 flags at the North Pole. You know you're in the right place. This is definitely the shell gene. So that was extremely satisfying. And you can see that they differ also in their geographical location. So more or less one of them is an East African allele, mostly from Tanzania and as far away as Madagascar, actually. Whereas the other arose in Nigeria, we think, but ended up in Angola and Congo as well. So they made a nice geographical distribution. Okay, so what is the shell gene? Well, it encodes something that we're very familiar with at Cold Spring Harbor. These things are transcription factors. What that means is they encode proteins that bind DNA and regulate other genes. So they're master genes, if you like. And that makes sense because it's a really important gene that clearly has major, major effects. It's a class of genes that's well known in other plants, especially the model plant Arabidopsis, where the gene is actually called seed stick, which you can almost tell it's got to be the right gene just from the name. And what was really nice about it was that the 2 mutations that we found were found in the DNA binding domain, which is where we would like them to be, and they influenced the interaction with another related protein called sepalata, which we were able to demonstrate using something called yeast 2 hybrid. You don't have to remember any of that. but it was extremely satisfying. And the reason it was satisfying was because it provides, at least for this example, an explanation for single gene heterosis. And that's because these 2 proteins interact with each other, and they need to interact with each other in order to bind the DNA. The DNA is this double helix around here. Here are the proteins, which actually form a multimeric sort of complex of these proteins. And that need to form a complex with other proteins formed the basis of a theory from an old friend of mine called Jim Birchler, who'd come up with this many years ago, where if you do the mathematics, if you need 3 blobs to be functional, okay, blue, red, blue, then if you vary the dose of either of those components, you will end up with something where the heterozygote can actually look better than either homozygote. This is because of dose dependence. So this really was wonderful scientifically because, first of all, we found the gene, and secondly, it had this wonderful explanation for single gene heterosis. But of course, most importantly, what this gene does is it increases yield. It means that we can now use this as a genetic test, just as you would any other genetic test, to find the trees that are going to have the biggest yield in any breeding scheme. And that will make a huge difference to the oil palm breeding industry. And so they were very excited about that. One reason they're excited about that is because they can take these elite hybrids, so they use the marker gene to find the best tree, just one tree maybe, not one, but say a few hundred trees that are elite, and they can take them and clone them. They can generate thousands of copies of the same tree by using something called micropropagation, in which they take what amounts to the heart of palm, if you like, which is the leaf at the top of the palm tree, and they take that, put it into tissue culture, and can regenerate literally thousands of little seedlings from that one parent palm. And because they're clones, they all have exactly the same genetic constitution. They've both got the 2 copies, the shell gene from one parent and not from the other. And so they should, in theory, have fantastic yield. And often they do these days. But sometimes they don't. And in fact, the first time this was tried, before it was perfected, some aberrations arose. And those of you who follow the cloning literature and animals shouldn't be too surprised at that. Of course, famously, Dolly the sheep was not quite the paragon of sheep health that she could have been, and her clones suffered from a number of these epigenetic maladies that even though they were genetically identical. And the same is true in palm trees. We often find problems like this where the fruit fail to form properly. These are actually parthenocarpic. They're very interesting phenotype in many ways, but they don't produce much oil compared to the normal ones. And this is bad news because you don't even see these fruit until the trees are about 10 years old, which means, of course, you've already planted your plantation, and again, you know, you've occupied a lot of land that you didn't want to occupy. And so the next thing that Orion Genomics is trying to do is use epigenetic technology, actually much of it developed here at Cold Spring Harbor, to find the responsible culprit for this effect. Okay, so palm trees then, oil palm trees are a very effective way of harvesting oil from the land, but of course there's a problem, and that problem is they coexist with the rainforest. And this picture was actually taken by Mylena, one of the oil palm team, from a helicopter above Borneo, and you really get the idea. Here are the palm trees being grown in uniform plantations, and here's the rainforest. So, you know, increasing the yield of oil palm is all very well, but we first, and I think it has great potential to, you know, restrict the expanded use of land, but of course, because of course you can now produce more oil from the same amount of land. But in order for that to work, you really need, you know, governments to work hand in hand with biology and make this work. And the Malaysians, to their great credit, actually put a moratorium on expanding into the rainforest any further in the 1990s, and the Indonesians have just recently done the same thing, about 2 or 3 years ago. However, you know, this is a really important area, and we all know why. I've actually spent quite a bit of time in Borneo myself, and I just have to introduce you to this guy, who sadly has passed on. This picture was taken 4 years ago, and I read recently that he died of old age. But this is Richie. He's the alpha male in the orangutan, or was in the orangutan sanctuary in Santubong in Borneo. And actually, I have to say, it's not a bad life for an orangutan in this particular sanctuary. It's, they get fed and so on. But the reason they have to have these sanctuaries, I'm just going to show you a few more pictures, but he's got amazing hair. It goes all the way in. Is of course because even with, you know, these rainforest, conserved rainforest areas, it's difficult for the orangutans to move around. And actually what's worse is that there are poachers who will attempt to steal the babies in order to sell them as pets, which is actually probably their biggest threat right now. But they are marvelous creatures. So, you know, rainforest conservation, you know, has to go hand in hand with increased oil yield. But oil yield increase through biology does have the potential to reduce rainforest impact. Needless to say, this has been an issue for a long time, and it was very interesting to see the reaction of the popular press to the genome of oil palm. I have to say, in general, it was very positive. The BBC World Service gave us lots of coverage, which was terrific. And even more surprising, as a Brit, was The Guardian, who you know, have really had a lot to say about oil palm plantations over the years. And we really got great coverage. It's very balanced and very reasonable. And even now, I mean, I think The Guardian produces an article on oil palm every few weeks, it seems. And they've really been investigating, you know, this issue, this idea that maybe by regulation and biological improvement, you know, we can really do something realistic with the system. I mean, you have to realize that most of the rainforest that's being destroyed in Southeast Asia is being destroyed by smallholders. And the reason for that, these guys are poor, and they need predictable yields in order to maintain their plantations, and without that, you know, the temptation to burn down, accidentally of course, the nearby rainforest is overwhelming. And if we can provide them with seeds that will always give high yields, then perhaps that will make a difference to that practice as well as the law. I mean, it is illegal. But anyway, yeah, the debate is one that's still very much going on. Given that, and also given another issue that you'll find raised here too, is another big problem with biofuels. And this is something that has been realized, especially with the production of corn ethanol over the years. And that is that corn and oil palm are foods. And actually providing food is as important as providing fuel, perhaps more so given the increase in the world population, which is predicted to go up to 9 billion by 2050. And so if you have to sacrifice food in order to get fuel, a lot of bad things happen. The first thing that happens is economic, of course, which is that the price of that food goes up. And that's exactly what happened with corn ethanol. When corn ethanol became much more widespread in its use, the price of corn went up quite dramatically, and there were riots in Mexico and so on, for very good reason. I mean, you know, this competition is a bad one. So although we've done a lot, I think, we have a lot of potential to improve the situation of oil palm with respect to the rainforest, we're not really going to be able to deal with this biofuel problem unless we deal with it in some other way. And I'm not saying that oil palm's not going to be used for biofuel. It is, and part of the reason for that, as actually Dagny was introducing before, is that the oil that comes from the kernel, the inside the shell, is actually mostly of a type that's better for is saturated. So it's better for biofuels and plastics and detergents and all that sort of thing than it is for food. So to some extent, oil palm is a versatile crop that can do both. But what we're really looking for is a plant that we can use for biofuels that is not going to compete for food. And so I just want to finish, and you heard Bruce saying something about this on that video, with one of my favorite plants at the moment, which I think has a lot of potential in this direction. It's called, you'll know it as duckweed. It'll cover your pond, especially if your pond is in a golf course where it gets lots of runoff fertilizer from the surrounding greens, because it loves actually to consume wastewater, and it cleans wastewater extremely efficiently as a result. You can grow it on municipal waste, you can grow it on agricultural waste. There are even mining companies in Australia that are growing it on mine water, which is quite remarkable because it can take up heavy metals as well. But the other thing about duckweed is that it grows incredibly fast. Oh yeah, just quickly, our friends at the American Museum of Natural History are placing it on the tree at the moment. They haven't quite finished yet, but we know it's going to end up somewhere around here. So again, it's a monocot. It's quite distant from palm trees, but nonetheless, we can sequence the genome just as we did the palm tree, and hope to get a lot of good information from that. And I'll tell you about that in a second. So here are the vital statistics. Duckweed, or most of the species, or lemna species, are in this family, Lemnaceae. They're the smallest flowering plants in the world. Funnily enough, they come from the same order, the Arcades, aroid plants, which are actually have the biggest plants in the world as well. So they seem to span the gamut. They're aquatic plants, which means they float, of course, on the surface of the water. That means they don't need all the lignin and all the heavy stuff that trees and corn have in order to be terrestrial plants, to stand up. They don't need any of that. So they're much softer, much easier to process, and they don't have a lot of the chemicals that make processing bad news. They bud, so they're clones. They bud fronds just from one another. If you put one of these things in a flask in the laboratory, they're quite scary. After a week or two, the flask will be full. And they really double incredibly fast, roughly every 2 days. And as a result, they actually have one of the largest biomass productions per hectare known. Even in industrial production, you know, commercially viable production, they're at least 2 or 3 times more than things like switchgrass and corn. And I don't mean corn kernels, I mean the whole plant. So these are producing more biomass than anything else. So they really have a fantastic potential from that point of view. There are several different species. I'm not showing them all here. The tiny little ones here are Wolffia globosa, which are, you know, you can get 5,000 plants into a thimble, which is quite fun. But although they grow clonally, they do produce flowers. And although they don't produce, and they can produce seeds, much of the, you know, some species without question, they also produce another sort of resting form, which they overwinter in. And these things are called Tyrions. You can see the dark here. And these actually contain almost pure starch. And so they have as much starch as a corn kernel does. So duckweed actually is a very promising biofuel just from the starch point of view. Think about sugarcane or something like that. But our goal at Cold Spring Harbor is to actually make them make oil, to convert that starch inside the plant, in vivo, if you like, into oil. And we think that's possible. We think that's possible for a variety of reasons. First of all, we've sequenced the genome. So just like the genome of oil palm, we've sequenced the genome of Lemna gibba, which is what's sometimes called common duckweed. And this is the form that it's everywhere. It's not invasive. It's normally found in a very wide latitude. And it's been used extensively in various types of research, especially in things like remediation. And so here are the biostatistics. We've got a nice genome. It's much smaller than the oil palm genome. I'm pleased to say it has a lot fewer genes, maybe half as many genes as oil palm. But it does have a lot of the genes that we think are going to be important. This is just an example. This is a gene required for oil biosynthesis. It's found in oil palm. It's found in other crops as well. It's actually one of my favorite classes of genes. It's a chromatin gene, a chromatin remodeler. But anyway, it controls, again, it's a regulated master gene that controls many others. So we're going to take a leaf, if you excuse the pun, or from the oil palm book, and see what sort of genes we can put in to duckweed to try to persuade it to make oil instead of starch. Now, in order to do that, you have to first of all develop the technology that allows you to put genes in. This is the genetic modification technology we were talking about earlier. And that's been extremely challenging with duckweed. A lot of people have tried in the past and have not got anything good, but I'm very delighted to say that a couple of people in the lab have been able to do this extremely efficiently now. What you're looking at here is that cell culture. Remember the callus that I showed you in oil palm? This is a very similar idea that's been transformed with a gene from jellyfish that gives you a green fluorescence. It's sometimes called GFP. And that made it very easy to find the transformed fronds, which were then selected, and these are very stable. The whole process is done within 6 weeks, which is comparable to some of our favorite plants that we use every day in the lab. And so this is really very good news. So we're very happy about this, and this is really going to provide us with the tools to make it happen. And these clones are very stable. This is a typical duckweed plant. You've got a little stipe here. You've got a thing called a pocket where the two new fronds are emerging. And you can just pick these things off, and they'll grow like the clappers. As I said, we're going to take a leaf from the oil palm genome and actually other oilseed genomes which have been looked at by others in the past. We understand a lot about shell gene, of course, but that's likely to be quite specific for oil palm because it has a shell. We're not dealing with seeds anymore. But what we are dealing with is genes that are expressed, and that's what these little graphs are, in the fruit, which is unusual for plants because it's a non-seed tissue and it's accumulating massive amounts of oil. So if we can take those same genes and put them into the fronds of duckweed, or maybe in the Tyrions of duckweed, then we should be able to make oil. At least that's the idea. And so this has been done at a sort of model level in other plants. Again, this is our favorite model plant. And it works reasonably well. We think we can get it to work even better than that, but that's using these very same genes. So these are some of our target genes, and I'm delighted to say I think we've got the first one in already, and so we should know fairly soon if we can at least at a sort of model level get duckweed to make oil, and then we'll have to really play with the conditions to get it to do it and grow fast at the same time. That's the hard part. But I'm confident we can give it a shot. Okay, I just want to finish on one climate change, last two slides, a climate change note. And I gave this talk recently at a summer school in Utrecht without realizing that much of the work that I'm about to talk about was done there. And so they were delighted. They came up to me afterwards, but it was interesting. So in the 1990s, actually, the Arctic Survey went out with icebreakers into the Arctic Ocean. In those days, you needed icebreakers in the Arctic Ocean. I'm not sure you do anymore. But they went out there and they drilled into the core, into the sediments, and pulled out cores, which they could then determine the plant life that had grown in the Arctic back to many millions of years ago. And what they found was that in the early Eocene, about 50 million years ago, they could tell from the carbon dioxide trapped in the ice that atmospheric carbon concentrations were much higher than they are now. And so the world was much hotter. The average Arctic sea temperature, for example, was about what it is in Long Island Sound, about 13 degrees centigrade. So there were palm trees and hippos in the Arctic Ocean in those days. But what was really interesting was that because of the plate shifts, the Arctic Ocean was actually partially landlocked. And so it had a top layer of fresh water after all that rainfall. And as a result, duckweed, actually a species called azola, was able to grow really, really well there. And they recovered something like 8 to 20 meters thick deposits of azola. Can you imagine these carpets of duckweed spanning the entire Arctic Ocean? This is an amazing biomass. And theoretical calculations suggest that this may have actually changed the climate, that there may have been sufficient carbon dioxide or carbon fixation to change the climate from the glass house that we had then to the relatively cool climates that we've been used to ever since. And so really these blooms of duckweed have some tremendous potential for climate change as well. Now, whether we could ever really seriously propose that is another matter, but these guys in Holland are actually working on the genome sequence now of this duckweed, azola, and we're in touch with them, and we're going to try and stay in touch with them about these projects. They have a fantastic video, which I don't think I have time to show you. I don't know, do I have time to show you? Yeah? Okay. Let's see it.
Present-day society is strongly dependent on fossil fuels. Not only for our energy needs, but also as a resource for countless household products. But fossil fuel reserves are finite, increase in cost, and affect our climate and environment. To maintain welfare for the growing human population, industry is permanently seeking alternatives. Resources that are sustainable, CO2 neutral, cheap, and practical to produce. Solutions can be found closer to home than one may expect, in nature itself. Plant material is particularly viable as a natural resource because plants use naturally existing nutrients, water and carbon dioxide. That's why in the pre-industrial era, wood and cotton were typically used as natural resources. More recently, soy, palm oil, rapeseed and many other candidates have substituted for fossil fuel, each with special qualifications in terms of quality and quantity. But all these alternatives came with downsides. They compete with food production for the use of arable land and require expensive and large amounts of fertilizers. All that is except for one. One plant species can change the entire ballgame. It is the aquatic freshwater fern azola.
I won't make you watch the whole thing, but you get the idea. So yeah, we're running out of time, but I recommend you have a look at this YouTube video. It's really fun, and they actually point out a lot of the industrial uses of the products you can get from these aquatic plants as well. So as I said, we're going to stay in touch with these guys, and hopefully we'll be able to make some progress on using aquatic plants as biofuels. Okay, well, we're at the end of our time, so I just want to give you some conclusions. So what I told you, oil palm is the most productive oil crop in the world, and we've been able to get to the basis of some of the hybrid vigor that's responsible for that production by cloning the shell gene. However, oil palm is also a food, and although you can make clones from oil palm, there are problems with cloning oil palm, which I think we can address. But ultimately, in order to maintain the sustainability, the balance between the rainforest and the plantations, we've got to consider other possibilities for biofuels. And I told you about our excitement about clonal duckweed and its successful, if very ancient, history in being able to solve some of our climate change issues. And so I'll leave you that food for thought. I've put up some names here. This is my duckweed group here at Cold Spring Harbor. Many of them, I think maybe all of them are here. Hopefully they'll be around to talk to you if you want to afterwards. The Oil Palm Genome Project was run by the Malaysian Palm Oil Board, and really the sequencing and a lot of the genetic sort of thought came from Orion Genomics, as well as our friends at the American Museum of Natural History, and also contributions from the Botanic Garden in NYU, which I didn't tell you about. And then thanks to Chef Smekens at the Azola Project in Utrecht, who gave me that video actually this morning, so that was good timing. and my friend Paulo Pereira in Brazil, who I talked to a lot about sugarcane. And so also the World Watch Institute provided much of the statistics that I showed you, and thanks to Cold Spring Harbor as well. I'd be happy to take some questions. Thanks.